Proteins are the most abundant organic compounds to be found in cells and comprise over 50% of their total dry weight. They are present in all types of cells and in all parts of the cell. Proteins perform many functions. They build many structures of the cell. All enzymes are proteins, and in this way they control the whole metabolism of the cell. As hormones, proteins regulate metabolic processes. Some proteins, for example, hemoglobin, work as carriers and transport specific substances such as oxygen, lipids, metal ions, etc. Some proteins, called antibodies, defend the body against pathogens. Blood clotting. Proteins prevent the loss of blood from the body after an injury, movement of organs and organisms, and movement of chromosomes during anaphase of cell division caused by proteins. Proteins are polymers of amino acids, the compounds containing carbon, nitrogen, oxygen, and hydrogen. The number of amino acids varies from a few to 3,000 or even more in different proteins. Amino acids. About 170 types of amino acids have been found to occur in cells and tissues. Of these, about 25 are constituents of proteins. Most of the proteins are, however, made of 20 types of amino acids. All the amino acids have an amino group NH2 and a carboxyl group COOH attached to the same carbon atom, also known as alpha carbon. In the general formula for amino acids, R may be a hydrogen atom as in glycine, or CH3 as in alanine, or any other group. So amino acids mainly differ due to the type or nature of R group. Amino acids are linked together to form polypeptides proteins. The amino group of one amino acid may react with the carboxyl group of another, releasing a molecule of water. For example, glycine and alanine may combine to form glycylylene dipeptide. The linkage between the hydroxyl group of carboxyl group of one amino acid and the hydrogen OF amino group of another amino acid release H2O and CN link to form a bond called peptide bond. The resultant compound, glycylylene, has two amino acid subunits and is a dipeptide. A dipeptide has an amino group at one end and a carboxyl group at the other end of the molecule. So both reactive parts are again available for further peptide bonds to produce tripeptides, tetrapeptides, and pentapeptides, etc., leading to polypeptide chains. Structure of proteins. Each protein has specific properties which are determined by the number and the specific sequence of amino acids in a molecule, and upon the shape which the molecule assumes as the chain folds into its annual, compact form. There are four levels of organization: primary structure, secondary structure, and tertiary structure. The primary structure comprises the number and sequence of amino acids in a protein molecule. Frederick Sanger was the first scientist who determined the sequence of amino acids in a protein molecule. After 10 years of careful work, he concluded that insulin is composed of 51 amino acids in 2 chains. One of the chains had 21 amino acids, and the other had 30 amino acids, and they were held together by disulfide bridges. Hemoglobin is composed of 4 chains, 2 alpha and 2 beta chains. Each alpha chain contains 141 amino acids, while each beta chain contains 146 amino acids. The size of a protein molecule is determined by the type of amino acids and the number of amino acids comprising that particular protein molecule. Now, we know that there are over 10,000 proteins in the human body which are composed of unique and specific arrangements of 20 types of amino acids. The sequence is determined by the order of nucleotides in the DNA. The arrangement of amino acids in a protein molecule is highly specific for its proper functioning. If any amino acid is not in its normal place, the protein fails to carry on its normal function. The best example is the sickle cell hemoglobin of human beings. In this case, only one amino acid in each beta chain out of the 574 amino acids do not occupy the normal place in the proteins. In fact, this particular amino acid is replaced by some other amino acid, and the hemoglobin fails to carry any or sufficient oxygen, hence leading to death of the patient. The polypeptide chains in a protein molecule usually do not lie flat. They usually coil into a helix or into some other regular configuration. One of the common secondary structures is the alpha helix. It involves a spiral formation of the basic polypeptide chain. The alpha helix is a very uniform geometric structure with 3.6 amino acids in each turn of the helix. The helical structure is kept by the formation of hydrogen bonds among amino acid molecules in successive turns of the spiral. B-pleated sheet is formed by folding back of the polypeptide. Usually a polypeptide chain bends and folds upon itself, forming a globular shape. This is the protein's tertiary conformation. It is maintained by three types of bonds, namely ionic, hydrogen, and disulfide SS. For example, in aqueous environment, the most stable tertiary conformation is that in which hydrophobic amino acids are buried inside while the hydrophilic amino acids are on the surface of the molecule. In many highly complex proteins, polypeptide tertiary chains are aggregated and held together by hydrophobic interactions, hydrogen and ionic bonds. This specific arrangement is the quaternary structure. Hemoglobin, the oxygen-carrying protein of red blood cells, exhibits such a structure.